Let's now look at the use of natural products in cancer therapy. So a variety of anti-cancer agents are actually derived from natural sources, and several of them are actually obtained from microbial sources, so they are antibiotics. Many of these anti-neoplastic antibiotics are produced by the soil fungus Streptomyces. Both the antibiotic and the natural product classes have multiple inhibitory effects on cell growth, and they primarily act to disrupt DNA function and cell divisions. And so that's the ones we start with. We have several mechanisms by which these agents target DNA, including intercalation, alkylation, and strand breakage, either directly or as a result of enzyme inhibition. And so what we'll talk about today is, for example, the idea of using intercalators. And intercalators is basically, it's generally caused by planar molecules, which has the right size, and these molecules will insert between adjacent base pairs of DNA, and in doing so, they cause a local unwinding that will disrupt the normal template function of DNA. So requires the drug to form both base pairing and hydrogen bond interactions, generally with a phosphate backbone. And so we'll find that many of those drugs have a flat aromatic component, and that's what we are seeing here, for example. And also they are positively charged. This ethidium bromide is actually not an anti-cancer drug. It's a dye that is used, for example, in molecular biology because it colors the DNA fluorescent very brightly. And so we use that in the lab when we are doing molecular biology experiments and we want to visualize DNA, for example, during cloning. In general, these intercalating agents are also very dangerous and they can cause cancer. A consequence of intercalation, and not the only way by which this works, is that in general, intercalators are also inhibitors of the topoisomerase enzymes, and there are several ways by which this can happen, and they can inhibit topoisomerase 1 or topoisomerase 2, and we'll see the mechanism more in detail in a little bit. So what does topoisomerase do? For example, let's look at topoisomerase 2, which is absolutely necessary for DNA replication. The way it works is that basically it binds upstream of the RNA polymerase DNA complex, and what it does, it removes the negative supercoil. So basically unwinds the DNA, allowing for the RNA polymerase to dock and copy. So the topoisomerase 1 makes a single strand break in DNA, and so allows the strand to spin and be processed. Topoisomerase 2, on the other hand, makes double strand breaks and allows an intact chain to pass through, and then subsequently, basically reattaches the two strands. Those enzymes are, believed to recognize sequences of DNA and bind, basically forming a transient phosphodiester bond with the backbone of DNA, and in doing that allows for breakage and reattachment. Okay, other natural products that we'll cover in this class inhibit the structure of the cell, or have to do with disrupting the replication process by disrupting the remodeling of the membrane during cell division. And we also have natural products that inhibit signal transduction, and we'll see several of them, some of which are natural agents and some of which are not, but we'll get started with that. So let's start our survey of natural products used in cancer therapy and that work as inhibitors of the topoisomerase. And for that, let's have a more detailed mechanistic look at topoisomerase. We have the supercoiled DNA. It binds to topoisomerase 2, which is a dimer and has an active site to tyrosines. We form then what we call a non-cleavable complex. So basically the state at which the DNA is associated with the enzyme, but nothing has started yet. And we have catalytic inhibitors that can work at this stage by basically disrupt catalytic turnover. Then we have topoisomerase 2 poisons that stabilize what we call the cleavable complex. And the cleavable complex, what has happened that we had strand cleavage of the DNA with covalent attachment of DNA to tyrosine. And then in the next step, normally the enzyme will undergo, with the expense of ATP, relaxation and religation. So, the DNA will be turned over and relaxed. And that allows then for copying DNA and also to synthesize RNA on the DNA template. So we have these two levels of attack of the enzyme. One is just inhibition, and the other one is stabilizing this complex with the DNA strand breakage, and that causes in turn apoptosis and cell death. We have two compounds here that are thought to work that way. One is amsacrine, which is actually used in research and not so much in therapy. But the idea is the same. They intercalate in the DNA, alter the shape, and therefore work as topoisomerase 2 poisons. And then we have a bunch of antibiotics, so natural products, for example, the actinomycins, that are isolated from various species of streptomyces. And they were initially investigated as antibacterial agents because the same mechanism of inhibition of topoisomerase 2 will also be antibacterial. However, these compounds are too toxic and so are reserved for anti-cancer therapy. How do they work? Well, we have a flat aromatic ring that intercalates in DNA and particularly prefers G-C pairs, right? And then we have attached to this ring, we have two cyclic pentapeptides in which we have non-canonical amino acids. So this is a non-ribosomal synthesis. And we have the formation of lactone between the C-terminus of the methylvaline and the side chain here, the oxygen of threonine. And so what happens is that the pentapeptides help anchoring the antibiotic to the DNA and reduce and increase activity. So the inhibition of topoisomerase 2 occurs by stabilizing the enzyme DNA complex. And so we can see this strand breakage. There is also resistance to this kind of antibiotics through the usual two-tier mechanism. One is reduced incorporation in the cell, so in reduced uptake, and the other one is increased efflux. And so the main actor in that is the P glycoprotein that mediates efflux from the cell. Similar to that, we have other antibiotics. And so those are topoisomerase 2 poisons. So what they do again is they intercalate and stop, stall basically the complex when the DNA is already cleaved. So same concept, they have a flat ring, and then in this case is a sugar that helps with binding to DNA and stabilizing the complex. In general, these anthracycline are also found from streptomyces, and there are 5 of them depicted here are used in therapy in the United States. What else? The main problem with these antibiotics is that they are cardiotoxic, and that's because they form oxygen radical through redox cycling. Other things about these antibiotics is that they're used in particular for non-Hodgkin's lymphoma, and they're relatively toxic. Here's a picture of these antibiotics intercalated into DNA, and you recognize here the sugar with the primary amine, particularly here sticking out. And remember, the backbone of DNA is negatively charged, and so having that positive charge helps with honing the antibiotics. So the planar aromatic chromophore intercalates, and the sugar sits in a minor group and interacts with the flanking base pairs. And so what it does again is prevents the detachment of the topoisomerase 2 and stabilizes the complex, and so the DNA helix cannot recycle. So as we say, the mechanism of toxicity is due to what we call redox cycling. That is the fact that we can oxidize the ring to form a radical, and then in the presence of oxygen we release a bunch of superoxide, iron 2, and that needs to be basically taken care of by catalysis. But so we generate a bunch of potentially toxic intermediates. And the important thing, why is the heart muscle particularly susceptible to that, is because myocardial cells tend to possess lower levels of catalases, and so they have less ways of getting rid of these highly reactive intermediates. And so other antibiotics that are used for inhibition of topoisomerase 2 are Cipro, and which is also basically an antibiotic, and it's used in particular against anthrax. And it works in the same way by inhibiting, in this case, the bacterial topoisomerase 2. And so topoisomerase 2 is also called gyrase because again it gyrates the DNA. We also have topoisomerase 1 poisons, and those are camptothecin, topotecan, and irinotecan, which work by a similar mechanism, but now they inhibit or they poison topoisomerase 1 instead. And remember, topoisomerase 1 is involved in remodeling single-stranded DNA. So with that, we get to another antibiotic, bleomycin, which is used in therapy, and it's actually very important at ESU because one of my colleagues, Sid Hecht, has done a lot of work on this antibiotic, trying to figure out the mechanism of action and also trying to make analogs that work better. So the antibiotic was discovered in the mid-60s from cultures of Streptomyces verticillus. It was put in the market in the late 60s, early 70s. It was also marketed by Bristol Myers initially, and it's used in the treatment of Hodgkin's lymphoma and other cancers. How does it work? Well, this is a very interesting mechanism. Bleomycin is a very complex molecule in which you can recognize some amino acids, for example, being linked together in weird ways, and that's okay. But what I'd like you to consider is that we have glycans here, which are in charge both of uptake and also they're in charge of helping with solubility and binding to DNA. Another portion here, which is positively charged and then kind of flat, binds to the minor groove of DNA, and then the activity is actually given by coordinating iron. And so in this resting state is iron 3 that gets reduced to iron 2, and in the presence of oxygen we get this highly reactive species which is formed, and that's the iron 4 bleomycin. And then bleomycin works as described in the slide by basically forming radicals on the ribose sugar, and that in turn results in strand cleavage and opening of the DNA, and so we form these single-stranded cleavage sites and these lesions that are very difficult to repair. And again, it's a radical based mechanism that requires the presence of iron. Another class of natural products that interfere with cell replication and are used as anti-cancer agents are actually acting on microtubules. Those are dynamic structures, protein-based. They undergo continual assembly and disassembly, and together with actin, they determine the cell shape, and they are involved in moving the intracellular movement of organelles. They also are involved in forming the mitotic spindles, so they play a very important role in cell division. They basically appear as these hollow tubes, and they're assembled from tubulin, which is a dimeric protein here formed by subunit A alpha and beta, and again self-assemble in these structures, which also present a polarity, which is how basically we can organize them in function. So we can interfere with their formation, and for example, these inhibitors for periwinkle, so from vinca, work by this way. So they're vinblastine and vincristin, and they're both used in cancer therapy. They prevent polymerization of these microtubules, so they prevent cell division. And the mechanism is again by binding at the termini of the tubule and interfering with the self-assemble. So they're very potent agents, and they're extensively used in several kind of therapy. The toxicity can be at the nerve level, because it turns out that microtubules are very important in nerve fibers, and so disruption of this function results in neuropathies that can be very severe. And another very important class of the agents that work on microtubule remodeling is Taxol. Taxol is one of the main treatment for breast cancer, and is also used to treat patients with lung and ovarian cancer. So it basically was initially extracted from the bark of a tree, Taxus brevifolia, but then turns out that it's actually a fungi in the bark that synthesizes this paclitaxel, which is good because initially, people were trying to prepare this drug by cutting the bark of the plants and killing the plants. So initially was developed at NIH, and so the supply wasn't the huge problem. We now have a semi-synthetic route to produce it, and that's very important. We also form it by direct fermentation of the cells discovered by this fungus, and that has allowed enough in the preparation of enough quantities to be used in therapy. So again, now it's produced by fermentation. Okay, so which one do we use in therapy? We say Taxol. Again, Taxol stabilizes the microtubules, and so basically gets the cell stuck. And here is a figure of Taxol in complex with tubulin. What else about Taxol? Well, one of the problems with Taxols is that it can only be administered in IV infusion. It's difficult to be solubilized, and so that makes the infusion relatively painful. and that's a problem, of course, for the patient. Other, a more recent way of delivering Taxol is to use these albumin nanoparticles, and so that was approved in 2012, and that has greatly improved the tolerance of Taxol by the patients.